we're going to start off on a little bit of recap. Not really recap, but kind of just adding on. Wait, what is it? Yes. Okay, there we go. Physiology. So this is just a kind of additional bit to the homeostasis and acid base that we covered last week. It's not very long. It's just one slide that we wanted to just quickly cover. So the normal body temperature is 37 degrees Celsius. Yeah, so it is also kind of a range. Like it can range from 36.6, I think, to 37.4. Yeah, so it's kind of anything between those two numbers is decent. And also neutral pH is 6.8, but the normal pH of the extracellular fluid is 7.4. So it's a little bit alkaline, a little bit basic. You know, all us humans are a little bit basic, you know, at some level. Yeah, but the normal range is 7.36 to 7.4. So as I said, a little bit basic. Anything above 7.4 is called, above 7.4 is called alkalosis. So that's basically your blood is too alkalotic, too basic. And anything below that is acidosis. Yep, so just remember that pH is does go up in log intervals. So there are bigger, they're like factors of 1,000 or something like that. Yeah. Year 12 chem, hell yeah. Yeah. So if neutral pH is 6.8, but normal pH is 7.4. So there is a slight difference between what is quote unquote neutral in daily life versus what humans actually are. Stop it. Yeah. Just a note for stuff in the body. You probably recognize this equation from chem or bio or something, probably chem. And this does happen in the body as well, not just in the natural world. So yes, so it's like when you're inhaling CO2, this will happen because you have water in your body. So like if you have too much CO2 in your body, you will increase the acidity of your body. And if you have less CO2, then you have less acidity in your body. Yeah, that's basically it. So central dogma, the one that we all love, and you know, on some level find very annoying. We're going to start off with the basics. What is DNA? So DNA is basically the coding for life. You know, the main things is it's universal, so all animals have it. It's basically evidence that we all come from a common ancestor. So thus we've all developed DNA and stuff in the same way. It is comprised of nucleotides. So one nucleotide is this, and which is one of these. And one nucleotide contains the phosphate group, the pentose sugar, which is that, and one of 4 different nitrogenous bases. This will be in the next slide, but this bit creates the sugar phosphate backbone of DNA. Because as you can imagine, sugar, phosphate, backbone. Yeah. So the bases, there are 4 nitrogenous bases. We have adenine, thymine, cytosine, and guanine. You probably would have learned this in bio if you did it. And if you didn't, here we go. Yeah. There's also uracil, but that doesn't come in DNA. That kind of comes in with your RNAs. Yes, so A and T are complementary, and C and G are complementary, as you can see in this diagram here. Yeah, so see, A and T is always together, and C and G are always together. Yep. And they will never bind with anything else except for those. Now, you've probably heard the words 5' and 3' floating around a lot in regards to DNA. And I will be honest, it took me a while to understand this too, so it can be a little bit, because they kind of expect you to know what they mean by 5' and 3' and accept it. It's like, excuse me, no, what the fuck is happening? But yeah, so basically DNA is directional, because as you can see from let's just go back for a bit. As you can see from a nucleotide, obviously there is a quote unquote direction to it. Like you have the phosphate side and you have the sugar side. And depending on where you are, like one side is 5' and one side is 3'. So this is how they're numbered. So if you think back to say organic chem, you have the way you would number your carbons in a compound is like 1, 2, 3, 4, and 5. So that is pretty much the basis for the 5' and 3' end. So the 5' end is the side that attaches to the phosphate, and 3' end is the side that kind of continues down and connects onto the next phosphate. Yeah, so that's what 5' and 3' means. So this is asymmetry, and it gives it a direction. Now, a very important thing to note is that DNA/RNA, depends which one you're doing, is synthesized in a 5' to 3' direction. So the way you can think about that is when, if you can think back to last week, when you have the, actually I might have a picture somewhere that I could just use. Yeah, so when you have like, so this is your DNA, this is skipping forward a bit, but if you were making RNA along that, this would be the 5' end, and you would be adding nucleotides onto the 3' end as you go. Yeah, so think about it as you're adding on to the 3' end. So therefore it is being produced from 5' to 3'. But then if you have to think about it, that means the DNA, if that's being made from 5 to 3, this is going to be 3 to 5. So the DNA is being read, quote unquote, from 3' to 5'. So RNA is synthesized from 5 to 3, DNA is read from 3 to 5. That's just something you kind of have to memorize, but it's very important to remember. I mean, you can kind of think of it like you add it to this end, like it kind of looks correct if you add it to that end rather than that end. I just thought I'd slide in here and say something about DNA mutations. This will come into play a bit more once you start genetics in a couple of weeks, but it is kind of a little bit relevant to, you know, DNA and nucleotides and stuff. So there are a couple of different mutations you can have. So there's point mutations, which are just a single base change in the DNA sequence. And there are a couple of different results of a point mutation, basically. So if it's a silent mutation, it's basically you've taken out, say, I don't know, say you took out an A and replaced it with a G. This would, you know, it would be changed, but it wouldn't actually change the phenotype. I've realized I probably should have put this in a little bit later, but let's just zoom forward a bit. So when you have your codons, as I think we might have touched on at some point, these are your mRNA codons, which is what's produced after you go through transcription. So say if you had like CUU, so if you convert that back to DNA, like GAA, and that A was replaced by another G, that would change it, but in the end you would still get leucine. So realistically, you haven't actually changed anything. But that's silent mutations. Yeah, so it could have, so the same amino acid was coded, or potentially it could have occurred in an intron. And introns are non-coding bits of the DNA sequence. So if the whole DNA sequence, only some bits are actually coded, and introns are generally cut out and just yeeted. So if it occurs in an intron, it doesn't really matter. Yeah. Then you have missense mutations, and that will actually change the amino acid and thus altering protein function. So you chuck something in and it went from like, oops, sorry, say CUU to CCU, and suddenly it's no longer leucine, it is whatever that one is, proline or something. Nonsense is if it codes for a premature stop codon. If you think about it, the way I like to think about it is nonsense has no in it, which is like just, no, you can't keep going, so it just stops it. And basically the protein produced is either like no viable protein is produced from that. And then there is frame shift mutations, which is basically like one whole base was taken out. So all of these ones have had one, you know, added in or replaced. And this has just inserted a whole ass thing. There's a question. Oops, sorry. What the shit? Change nucleotide into complementary nucleotide. Okay, so we've got a question that is, would these mutations change a nucleotide and its complementary nucleotide, or would just the single nucleotide be changed and produce a mismatch? I would say this, you can't really have a mismatch. Like, I don't think the forces would actually like allow that to happen. And usually, I guess this is kind of more, well, when mutations happen, it kind of, I would say it would affect both the nucleotide and the complementary nucleotide. The reason why we only talk about kind of one strand is that, you know, when you're transcribing it, or, you know, DNA synthesis or something like that, it will just kind of go off the one template strand, but realistically, it would be both nucleotides. Cool. Yeah, so when you have a point mutation, it can lead to loss of function, gain of function, or conditional mutations. And this will become very important later, especially when you're doing cancer, because some mutations can cause loss of function of cell cycle, gain of function cell cycle, etc. It's like when P53 is mutated. I think we've covered that. Have we covered that? Potentially. Do we know what P53 is? If we don't, well, we'll get to it. And P53, I mean, I guess in brief, is basically a gene that codes for regulation of the cell cycle, and when it's fucked, the cell cycle is fucked. So that would be considered a loss of function mutation. So I'm quickly going to touch on proteins. So proteins are made from subunits called amino acids. So like one of these circles is an amino acid. And there are 20 different amino acids, but these are coded for 64 different combinations, as seen on this table. Because you can see, as you can see, multiple different combinations lead to the same amino acid, hence 20 and 64. One amino acid is coded for by one codon, and a codon is a group of 3 nitrogenous bases. And yeah, different combinations can code for the same amino acid. A chain of amino acids, which is produced after protein synthesis, is considered a protein. So that's the most basic structure of a protein. And as the proteins fold and, you know, interact in different ways, they become more and more complex and more folded and become more complex substances. So for example, like quaternary protein structures can be like hemoglobin has a quaternary structure, and tertiary protein structures, enzymes have tertiary structures. Whereas a primary chain is just literally a chain. So yeah. So we're going to move on to transcription and translation. So this is basically how proteins are made is summary. It's basically protein synthesis. And the two parts are transcription and translation. So in brief, your DNA is transcribed into RNA, here, RNA. And then the RNA is translated, quote unquote, into a polypeptide chain or a protein. So that's kind of the overview of what we're going to get at. So transcription. There are a couple of products of RNA transcription, and they can be mRNA, rRNA, or tRNA. tRNA is, I mean, it's oh my God, I can't speak, mainly mRNA. And that specifically codes for proteins. So that's kind of what we're going to be focusing on today. But tRNA is also important in protein synthesis because tRNAs are what carries amino acids to the ribosomes. As we lightly discussed last week, you have your ribosome and like things go through it. We will, elaborate on that in a second. So different RNA polymerases are responsible for different activities. So RNA polymerase 2 is responsible for mRNA synthesis, but at the same time, RNA is not proofread, and so an error can occur every now and then. So transcription, this is, I'm fairly sure, yep, this, I guess, simplified version of transcription. So transcription is a synthesis of an mRNA strand that is complementary to a segment of DNA. So this particular segment of DNA that we have is like, so you have the long strand of DNA, we just want this bit, because hey, this one bit codes for the protein that the body wants to express. So also think about it as the body wants the protein, say like, I don't know, insulin. So then it'll trigger the cells to produce insulin, and this is what happens. Yeah, so first off, the DNA is unzipped by helicase, which separates two strands. Oh, sorry, there's a question. Okay, Stella's got it. We're all good. Thank you, Stella. Yeah, so DNA is firstly unzipped by helicase, which separates the two strands. And then you have an enzyme called RNA polymerase. And that's very similar to DNA polymerase, but it's for RNAs instead. So they bring in RNA nucleotides. So these are very similar to DNA nucleotides, but the key thing is that RNA has uracil rather than thymine. And uracil is complementary to adenine, as T is. So basically anything that was, any T's are instead used. Yeah, so they just kind of go along and bind to anything that is complementary. Then the mRNA detaches from the template strand and leaves the nucleus via nuclear pores to go to the ribosomes for translation, and the DNA is bound back together. This is a very important note, because this came up in our exam and really screwed a lot of us over because of the finicky wording. So, do I have a diagram? I have a diagram. So if you look at this, you have a coding strand and a template strand. So the coding strand is just, the accessory one, and the template strand is like the one that the mRNA is actually being copied off. The thing with this is you have to remember that the coding strand will be basically the same as the mRNA that is being produced. The sequence of bases will be the same, except T's instead of U's. So as you can see down the bottom here, you have your coding strand and your template strand. So your template strand, as you can imagine, template strand, you can imagine is the template for RNA to be produced, and that's complementary to the RNA. However, the coding strand is pretty much the same. It's exactly the same, except T's are now U's. So they can ask you a question where it's like, this is the sequence of DNA on the coding strand. Tell us what the mRNA strand will look like. And a lot of people made the mistake of thinking it's a template strand and writing out the sequence as if it was complementary. But it should actually be the same. So just keep an eye out for that. I guess a little bit more in depth for RNA transcription, you can think of it as happening in 3 stages. So there's initiation, elongation, and termination. So in the initiation, RNA polymerase, that's that blob there, it comes along and binds to a promoter site. So promoter sites are basically these areas on the gene that, they don't really code for anything, but their particular sequence is like the recognition site for the RNA polymerase to bind to. So the RNA polymerase will bind to the promoter site because it's like, oh, I recognize this. Okay, I'll go over here. So it comes over there, and that separates the 2 DNA strands. Then you have elongation. That's this bit. So elongation is, I guess, the elongation of the mRNA strand. And as the RNA polymerase moves along, the mRNA strand is produced. And then there's termination, where a termination site is reached, the polymerase goes, oh, okay, we're done. And the mRNA strand will just, stop being produced, and it's time to float out of the nucleus and go into the cytoplasm, where we find ribosomes. I thought I would just quickly touch on transcription factors, because I remember we kind of learned this, and then they were like, yeah, teach yourselves. And we were like, what do you mean? So these are transcription factors, and it's a very broad term, transcription factors, and they mean a lot of different things. Like all of these little blobs are transcription factors. They're all slightly different in structure, but they all basically have the same role. So yeah, transcription factors are proteins mainly, and they just aid transcription processes. So you have the promoter site here, right? There's a promoter site and an enhancer site. So if that is the promoter site, this pink bit is the enhancer, see? And basically it just, when you have transcription factors, they will want to come along and kind of attach onto the promoter site. And that becomes like a bigger beacon for the RNA polymerase to see. So the RNA polymerase will come along and be like, oh, I see. There's where everyone is. So it'll come along, go into that. And then the transcription factors make kind of a bridge between these two sites. And what that does, it curves the DNA around. And I'm not gonna lie, I'm not really sure why, but this makes it easier for the RNA polymerase to transcribe the material. Yeah, because I think it just brings the two brings the strand like closer together, so it doesn't have to like, I don't know. That's basically what they tell us, and that's basically all you need to know. There's the enhancer and the promoter, and you have a bridge of transcription factors and RNA polymerase, and it forms this kind of curvy bit, which makes it easier to transcribe. Yeah. Oh no. What the shit? Okay. Repressors. Just a quick note, I guess. Repressors are regulatory proteins that act to turn off transcription. And I guess they're kind of similar to TFs, but the opposite. So yeah, they may function by competing with activators for binding sites. So instead of the TFs binding, these bind instead. They can mask activator activity or just, having a negative interaction with the transcription complex. Yeah, so they basically are these proteins that are little shits and interfere with stuff. And I guess it's just sometimes if, the body's like, this is enough, we don't need any more protein, it'll just send these things. Yeah, so I thought that we'd also touch on RNA processing. So we can't just let the newly synthesized mRNA just leave. Like, that would be irresponsible. We can't just let it leave. Before it can leave, RNA must undergo some additional processing. So firstly, a 5' cap and a poly A tail are added. So you can also call it a methyl G cap and a poly A tail. So basically, a poly A tail is a bunch of adenines. As you can imagine, poly A, many A's. And then the 5' cap is basically just like a guanine that's tucked at the end. And these two things basically just protect the RNA from being damaged once it leaves the nucleus. So, there are a lot of big scary things in the cytoplasm. You never know what could happen. So those are kind of its buffer, its protection to like seal off the strand, because we don't want to lose any of this important information. And then there is RNA splicing. So you have introns and exons. So if you think about it, so introns are spliced out and exons stay. Yeah, so I guess a way, I'm trying to think of a way you can remember it. I guess you can think that exons exit the nucleus. So they actually make it out of the nucleus as part of the final mRNA. So exons end up exiting the nucleus, whereas introns stay in the nucleus. Yeah. And this splicing occurs via spliceosomes. Very creative name. And they are formed by snRNPs. Don't question it, just accept the name and move on. And they bind on either side of an intron and then meet together and then yeet that boy out, basically. Okay, I've got a quick question just to make sure everyone's still paying attention. So the base pattern of the coding strand over length of DNA is as follows. And what would the mRNA strand from this length of DNA look like? So I will just sit here and wait to be able to reply. We have a couple of C's, and that is great because it means that you did take home the message I was trying to aggressively say earlier. Sorry about that. But yeah, no, that's really good. It's good to make sure you read whether it says coding strand or template strand, because a lot of people were tripped up by that because no one really, made a point of it. Yep, so that's just the summary again. Question 2. What direction is the template DNA strand read in? Yay, B. That is correct. As you can remember, the RNA strand is produced from 5' to 3', and this is moved along from 3' to 5', as you can see. Coolio. Now we're going to move into translation. So your mRNA has now left the nucleus and it's making its way to a ribosome. And often the ribosomes will be on the rough endoplasmic reticulum, as we touched on last week. And the rough ER is studded with ribosomes. So that is where we find ourselves now. Ribosomes. So as I may or may not have mentioned last week, but there are basically 3 subunits within a ribosome. And this is the A, the P, and the E sites. So first, A stands for the amino acyl site. And that's a bit like, what does that mean? Basically, it just means that, there is an amino acid that is bound to the tRNA that's coming in. Okay, on the basis of tRNA. So this block here is a tRNA. And the tRNA is basically a protein that is kind of floating around near the ribosomes. And initially, each tRNA has an amino acid bound to it. Yeah, so you have one bound to it there, and that will come in. Once it leaves, that's gone, but in the general body's cycle, it'll be replenished. Yeah, so that's amino acyl site. The peptidyl site, which is, you can think of it as the peptidoglycan chain is forming, not peptidoglycan, sorry, the polypeptide chain is forming. And then the exit site, which is, as it sounds, the place where they exit. So yeah, as you can see, they're also just in the opposite orientation. So like the mRNA moves through that way, but even though the subunits are kind of the other way around, if that makes sense. So it's in the order EPA, even though it goes through APE. Yeah, so the amino acyl site is where the incoming tRNAs bind to initially, and they have, their amino acid, and it's bound to, see, as you can see, the complementary codon. So AAG binds to UUC. And then kind of, at any given moment, you kind of have 2/3 in there. In the middle side, it will, basically, that comes in, and it'll shunt that one out of the way, and this polypeptide chain will transfer over to this one, and it will add on to here, and then it gets shunted back out again and yeeted out the ribosome. Yeah. So yeah, so the basics of translation is that, there are 3 mRNAs called a codon. tRNA molecules have complementary codons. As the mRNA and tRNA move through the ribosome, the amino acids bind together and form the polypeptide chain. And once the mRNA strand has been fully read, the mRNA and polypeptide chain are just released into the cytoplasm to be transported wherever they must go. Yeah, so that's what it looks like. So as you can see, as an overview of the whole process, you have DNA transcribed into mRNA, leaves the nucleus, mRNA travels into the cytoplasm via a nuclear pore, and they're just basically tiny holes in the nucleus, and it goes to a ribosome, and yeah, your protein is synthesized. Just to go into a little bit of depth, that's kind of what I just explained earlier, but I guess in slightly more scientific terms, then the tRNA is yeeted out of the ribosome. But yeah, it basically explains how an incoming tRNA binds to that, a peptide bond forms between that one and that one, and then this whole thing is shunted over here, and they bind together and form the chain, and then it gets yeeted and everything moves along. Yeah. There's the photo again. So, quick question. What is the order of the sites in a ribosome from left to right? Hooray. Yes, it is D. Wonderful. But do keep in mind that it moves through the other way. I also discovered while I was doing this that you can make a lot of different words from the letters P, A, and E. Anyway, side note. After translation though, sometimes you can have a couple of modifiers. this, I guess, kind of plays into gene silencing, which we are also getting to. So there are a couple of different ways that proteins can be modified after they have, after they've been like translated into the polypeptide chain. So most polypeptides must be modified after translation in order to become functional. So there are a couple of things that happen, and these are the main three. So you have proteolysis, glycosylation, and phosphorylation. And their names kind of explain what they do. So you have proteolysis, which is, if you think about it, protein, and lysis, cleaving. So it cleaves the polypeptide, and that allows the remaining fragments to fold into different shapes. Although sometimes this can lead to activation of an inactive precursor. So sometimes if you have like proinsulin, which is the precursor to insulin, it can be cleaved, and those remaining fragments can freely be formed into insulin. Because you don't want insulin to always be active in your body all the time, because otherwise you would, that's not very great. So therefore it's kept in proinsulin until it needs to become insulin, and then proteolysis happens. Then you have glycosylation. If you think about it, like glyco, it's like sugar, and glycosylation, anything with like ylation is always addition. So that's adding sugars, and that can often be important for targeting and recognition. So actually with the different blood types, you have, A, B, O, all that stuff. The reason why they're different is because they have different sugars. That's not really something you have to know, that's kind of just an example of how it works. And then you have phosphorylation. And phosphorylation is when you add phosphate groups to the protein, and that alters the shape of the protein and its interaction with other proteins. And just a couple of side notes, that phosphorylation is mediated by kinases, while dephosphorylation is done by phosphatases. Yeah, so dephosphorylation is obviously getting rid of the phosphate, and that's done by phosphatases. You can think of it as the phosphatases want to take the phosphate. They're like, phosphate's in our name, we want it back. See you later. Phosphorylation may affect protein interactions or activate switches within proteins that can result in activation or deactivation. Yeah. Sometimes certain modifications can only happen in specific compartments. Yeah, for example, glycosylation and disulfide bonds are characteristic of the endoplasmic reticulum. Yeah. I thought I would just touch on ubiquitination because on our Curious Cat last week, go check out Curious Cat if you haven't, if you don't want to ask a question, but want to, hit up Curious Cat. Ubiquitination is basically the targeted degradation of a protein. So basically Jesus Christ, the truck. Okay. Degradation of a protein is when it's like, it's done its job. They kind of want to get rid of it now. So they send these things called ubiquitin. So ubiquitin will literally just bind onto the protein. And once it forms a long kind of strand, it is called polyubiquitination. As you can imagine, polyubiquitin. Yeah. So this ubiquitin kind of acts as like a target for proteasomes. So the proteasome is like, oh, ubiquitin. So it comes along and it's like, oh, okay, this protein needs to be eaten. So it'll eat up the protein, break it off into smaller fragments, and basically, the protein is digested and put back into the body, whilst the ubiquitin is then released and can be reused. Yeah, so that's pretty much what it is. And that's just used, I guess, as a way to regulate what proteins are actually being used. So we're going to learn about gene silencing. And I added this bit about that one active learning that was really confusing and no one learned anything, because that's what happened for us. I don't really know if you guys got that active learning, especially because I don't know if you guys get active learnings at the moment. But basically there was this guy and he gave us this whole Moodle notebook of random DNA shit to learn. And we were like, each only had to learn one part and teach it to each other. And obviously when we taught it to each other, we had no idea what was going on. So I thought I'd go through that whole thing and talk about it a bit. Oh shit. So basically the summary is that gene silencing happens in a couple of different ways. And it starts, basically you can start from the DNA structure itself. It can occur in RNA transcription. It can occur in like RNA processing and silencing in that bit. and post-translational. So this is what we just covered, the protein stability and activity mostly. So that's kind of like this stuff. And then this stuff I'll cover as we go along. So to start off with the basic structure of DNA, if you think about it, if the transcription factors, if they can't access the DNA, then they're not going to be able to transcribe the DNA. So here you can see that in a normal cell, like even just normally your DNA is floating around the nucleus, you have your DNA that is bound up into basically chromatin. So the way chromatin is structured is you have your DNA helix that gets wound up and curled around a histone, and a histone is basically a protein, and each unit is called a nucleosome. A nucleosome is made up of 4 histone molecules and DNA wrapped around it. In between, you have your linker DNA, and it basically just keeps going and winds tighter and tighter and tighter until you have chromatin. Yeah, so as you can imagine, when it is tightly bound like this, you can't really like get an enzyme to get itself in there and translate some of the DNA, as you can imagine. So this in itself, like the physical structure of DNA, is the first level of gene expression. So I thought we have to talk about histone acetylation. So the way that DNA is kind of kept to the histones is by these tails that histones have. So histones generally tightly bind to DNA because they can bind better. Like just for packaging purposes, it just keeps everything closer together. So histone tails are positively charged. DNA is negatively charged. So when they're together, they'll interact, they'll want to bind tightly to each other, and that closes it off. So this DNA is inaccessible, and you can call it's inactive because you can't access it, and this is called heterochromatin. So inaccessible is heterochromatin. However, then you have histone acetylation. So when the tails are acetylated, it's basically you just add an acetyl group to the histone tails, and this releases it. And because this interaction is, weaker. So now your DNA is, it's slightly more accessible because it's not bound so like skin tight to the histone proteins. So now your transcription factors could come along, bind onto there, and, actually transcribe something. So this active chromatin is called euchromatin. I guess I like to think of it as euchromatin. I guess the first thing I think of is like euphoria. I don't know why. And I guess if you're happy, it's like you're active and you're happy. I don't know, that sounds like a really shitty, analogy, but you can come up with whatever you want to help yourself.
Another way to remember it, sorry, just jumping in. Euchromatin is used. So the first bit makes, I thought you knew that.
I genuinely forgot that.
Yeah, euchromatin is used.
That's much better. Fuck. Use that one, guys. Euchromatin is used. That makes a lot more sense. I forgot a lot of the helpful hints from last year, I'm not gonna lie. There's a lot of information. Now we're just gonna touch on how a nucleosome is modified. So that's the same thing. That's the same slide twice. Don't worry about that. So just have a look at this slide, which is basically how it works. So as you can see here, everything's really tightly bound. You have histone acetyltransferase, and that, as you can imagine, histone acetyltransferase transfers acetyls to histones. So as you can see here, all of these tails are not acetylized, and here they are because you can see the little circles. And now that it's loose, the remodeling protein can actually come in and access it. So now that that's kind of opened it up, it disaggregates the nucleosome, it opens it up, the histone has fallen away, and now the transcription complex has space to actually bind and begin transcription. Yep, and to deacetylize histones, an enzyme is called histone deacetylase is activated. They have really creative names like histone acetyltransferase and histone deacetylase. Wow. Yes, so histone deacetylase, gets rid of the acetals, and this causes DNA to wrap tight back around the histones again. And that condenses the chromatin, so then it's inaccessible. This is called gene silencing, as the genes can't be expressed. So sometimes even if it was kind of existing like this, if you just acetylated it to access the thing, once you no longer want the protein to be produced, it would be deacetylase and it will come back in together and you can't transcribe it anymore. So now we're going to talk about DNA methylation. So DNA methylation is the addition of a methyl group to cytosines, and it's only cytosines, which is kind of strange, but it is what it is. So basically, you have a cytosine here, as you can see, and in methylation, you will basically just attach a methyl group onto your sleeve, and it can happen there too. And basically, when you have that there, it just inhibits transcription of the gene. I guess one way to think about it is like, it doesn't affect the base pairing, so it's still bound to G. But if you affect it, so like, as you can see, different types of cells have different DNA methylation profiles, and if that's altered, then it can fuck it up basically. But methylation itself, I guess, you can't really transcribe something when you have the methyl group there. Like, it basically just interferes with all the interactions between like the DNA and the enzymes and RNA polymerase and stuff like that. So basically, once it's acetylated, it prevents transcription. Yeah. And if that's not done properly, it can contribute to human cancers and some diseases. Okay, we're going to touch on control by non-coding RNAs. So this is miRNAs, microRNAs, and siRNAs. So this isn't really that high yield, but it is something that they like to ask you questions on just because they know people will be very confused by it. But it's not actually that difficult, and they both basically do the same thing. So microRNAs are basically these short bits of non-coding RNAs that inhibit RNA translation. And these are very specific. They can be specific to certain tissues and certain stages of development. So the miRNA precursor is transcribed from genomic DNA and is cleaved by an enzyme called dicer. So it's kind of very similar to this one. So you have your RNA, that's your precursor. Dicer comes along and snips it up. As you can think about it, dicer literally dices it up. So the mRNAs are loaded onto the RNA-induced silencing complex. That's not really something you really have to remember. It's called RISC, and it is converted into single-stranded RNA. As you can see here, it's loaded up into there, and it's single-stranded. The RISC will scan the mRNA, and basically, because it is both RNA, so they both have bases. So at some point along the thing, these are very specific. So they will have a particular target that has a complementary sequence. So say I want to bind exactly there, because it's like if we bind there, then it will prevent transcription. So it comes along and binds to the mRNA and therefore prevents translation. Literally exactly the same thing happens with siRNAs. These are siRNAs because they are small interfering RNAs, which honestly, miRNAs are also small interfering RNAs, but aren't called that. I don't know, man, bio. And they literally do the same thing. Like exactly the same thing. You can find this on our document and it's literally basically the same thing. siRNA is cleaved by dicer, it's loaded onto RISC, converted to single-strand DNA, scans mRNA, and binds to its target. Yeah. And finally, we're just going to talk about mislocation and sequestration of proteins. So this is another way that you can control the activity of proteins. And basically, if you think about it, certain proteins have specific environments or specific compartments that they want to be active in. And if they're not in that compartment, they could be inactive because, the environment's not right or doesn't have the correct enzymes or proteins that they need to be active. So yeah, so each compartment of the cell has a specific environment, and that could be incompatible with the correct folding and stability of the protein. For example, failure to translocate into the endoplasmic reticulum will prevent formation of disulfide bonds that we kind of touched on earlier, and glycosylation. So therefore, the protein might not fold properly. Yeah, so you don't actually have to remember these examples exactly, but basically, if a protein doesn't make it to where it's meant to be, sometimes it might not function properly. And if it's in the wrong compartment, it could produce a toxic effect. So that's mislocalization. So that is kind of, I guess, almost an accident. Sequestration is quite purposeful, and it's sequestering a protein within a specific compartment where it doesn't have any effect, like whatsoever. So it's like, we have all these proteins, but we don't want them to affect it, so we're just going to chuck them in this corner so that they can't hurt anyone. Yeah, but then when conditions change, the sequestered protein is released into where they want it to go, and it can become active. Yeah, and this migration allows control of the protein. And yeah, well, that's pretty much all it from, well, no, that's not all it from me. I have a question. In histone acetylation, what happens to the DNA accessibility? Let's see if you remember which one's heterochromatin and euchromatin. Yay. And yes, the answer is A. Thank you very much. As you can remember, euchromatin is used. Thank you, Jess. And when you don't have acetyl, so the addition of an acetyl makes it more available. So you add an acetyl, heterochromatin becomes able to be used. Yeah. So that's it for me today. Now I'm going to be passing on to Xenia, who is going to be taking you through biochem and cellular respiration.
All right, hello everyone. So my name is Xenia. I'm going to be taking over from Izzy as she zooms off to class. So today we're just going to cover enzymes and cellular respiration. Next week we'll probably be covering all of metabolism. So I know you guys probably had one lecture on fuels, I think. So that won't be covered today, but will be covered next week. Just wanted to say something about metabolism. Everyone finds it really hard. Everyone kind of hates it. But the main thing is not to worry about it. It will all make sense at the very end when it all comes together. So you just have to hold out this week with a bit of, yeah, it's just very hard. So at the end, it will all come together. So don't worry about it. But today we'll just start with some enzymes. So this slide, so a lot of this, what I'll be covering today, there'll be a lot of definitions, some that you'll have to know and some you just have to understand the concept behind it. So this slide, where we talk about different types of energy and thermodynamics, you do not have to remember the definitions of this, but this is something that you have to understand. So as you would know from biology and chemistry, hopefully, there's a lot of different types of energy. So there's something called potential energy, which is basically the energy stored in chemical bonds. And this energy that's stored can be released and used, it can be converted into different types of energy. For example, kinetic energy. So kinetic energy can be used, for example, in enzymes. It can actually function, and that's how these conversions work. So it's just important to understand that there's just a lot of energy conversions, which you should already know. So with the law of thermodynamics, so basically you just have to know energy can't be created or destroyed. And you also have to know that sometimes it's not very efficient. So some energy is going to be lost in conversion. So a good example of this is thinking about heat. So as you know, the body's quite warm, and in reactions, it loses a lot of energy as heat. That's sort of an example. Again, you don't have to, you will not be asked in the exam, what is the first law of thermodynamics? But this is a concept you have to understand in order to basically understand the rest of metabolism. So we'll keep going with that. So calculating energy, you might have seen this in your lecture slides. All you have to know is that total energy equals usable energy plus unusable energy. And that sort of feeds into the concept again that not all energy conversions efficient, okay? And there's always going to be some energy that's lost. This bit here with the H equals G plus TS, this is just another way of showing total energy equals usable plus unusable energy, okay? You're not going to get asked this on the exam. This is more just something to understand. So this bit is important, and these are definitions that you do need to know. So exergonic versus endergonic reactions. In biology, we say endergonic and exergonic, whereas in chemistry, you would have seen it as exothermic and endothermic. They're basically the same thing. Biologists and chemists just decided to be different, I guess. So exergonic reactions are also known as catabolic reactions, and they're spontaneous reactions that release free energy. Okay, so how I like to remember this, it is going to sound kind of dumb, but the cat out of the bag, okay? So the cat is like the energy and it's being released, okay? And that's how I remember it as catabolic, because it has cat in the name. Endergonic and anabolic is just the opposite to that. So they're non-spontaneous and they require energy, so they absorb it. These reactions are generally involved in synthesis. So you can remember it as just the opposite of exergonic. Or if you want a way to remember it is if you've ever heard of anabolic steroids, which is what bodybuilders seem to abuse to get bigger. If you think anabolic steroids is getting bigger, you can remember anabolic means building up in synthesis, okay? So another important thing to remember is this triangle G. So this just means change in energy, okay? So if we have a reaction that's exergonic and it's releasing energy, it's getting more negative, okay? Because it's losing its energy. Whereas if we have endergonic, it's getting more positive because it's gaining more energy. So that's where that comes from. And how we understand that is how we figure it out is we look at the energy of the products minus the reactants. So this will make a bit more sense in the next slide. So again, this you don't have to memorize, you just have to understand, and you might get a diagram like this in the exam, you might not, it just depends, okay? Again, no memorizing, just understand. So if we look at this axis here, this is just looking at the amount of free energy, okay? This axis is just the time or the process of the reaction, okay? So if we look here on the left, so we're gonna first look at endergonic reactions. We have reactants down here and products down here. Okay, so remember in an endergonic reaction or an anabolic reaction, energy is absorbed. Okay, so you can see here, if we look at this blue line, it's getting bigger and bigger up until here. All right, and that's because energy is being absorbed. All right, if we look at exergonic reactions, which are also catabolic, you can see here we start with reactants. Then remember, energy is released. So from the reactants, they're going to have less energy. So they're going to fall all the way down here. Okay. Now, one thing this diagram doesn't actually show, which is really important a bit later for enzymes, is something called activation energy. So activation energy is the amount of energy needed to start the reaction, okay? And how we calculate activation energy is we look at the reactants down here and we look at the peak up here, okay? See that peak? So the activation energy is all the way from the peak to the bottom of the reactants, okay? So that's for endergonic. If we look at exergonic, okay, we look again from the reactants up to the peak. So in this one here, you can see that the activation energy for exergonic reactions is actually a little bit lower than for endergonic reactions, okay? That will make a bit more sense in the context of enzymes, so hang on, and we'll keep going. So I'm just going to do a bit of an overview of ATP, also known as adenosine triphosphate. This is a really, really important molecule. It's the energy currency of the cell, and we'll talk a lot about it. And you should probably actually understand what it is, because I know it's just really random. You're talking about it a lot and you might not know what it is. So basically ATP is a nucleotide that consists of a nitrogenous-based adenine, which is what you just find in DNA, the sugar ribose, and it also contains a chain of 3 phosphate groups. So if you can see this diagram here, we have our nitrogenous-based adenine, we have our ribose sugar, and we also have these 3 phosphates. Now, what's really important here is looking at these bonds between these phosphates, all right? So you can see here, it has a little bit of a label, a high energy bond. And these bonds, remember back to our very first slide, they contain potential energy, okay? And when we add a water molecule, it's called hydrolysis, you should remember this from chemistry, this bond is actually broken and it releases a lot of energy, okay? Then when you get rid of a phosphate, it goes from adenine triphosphate to adenine diphosphate. Okay, so that's also important to remember. ATP also has one more function that's not really talked about, and you don't really have to know this as such, but ATP can also phosphorylate many molecules. So it means it just adds a phosphate group, okay? All right. This is just a diagram here that just shows it as a more visual representation, okay, for those of you who like that. So again, high energy bond gets released energy. Here, adenine diphosphate doesn't have as much energy, okay? So we'll keep going. So now we're going to start enzymes, all right? So basically what an enzyme is, or firstly, we should talk about catalysts. So enzymes are just biological catalysts, and a catalyst is a substance that increases the rate of reaction without itself being consumed. Okay, and enzymes are just protein catalysts found in organisms, okay? So how they work is by lowering the activation energy barrier, okay? So if we go to the next slide, here's an example of what I was talking about earlier. Remember how we find out the activation energies? We look at the reactants, okay? And then we look at the peak of this diagram. And it's just that difference between the peak and the reactants, all right? So if we have a look here, the purple line is the reaction without the enzyme present, okay? Whereas the green one is the reaction with the enzyme, okay? And you can see here, the main difference between the two is that with the enzyme, there's a smaller activation energy, okay? So enzymes, they lower the amount required for activation energy, and how they do it is they provide an alternative pathway. And I know that's just a fancy word. Alternative pathway is just a fancy word for saying that sometimes they can physically fit different substances and enzymes together. They can physically rotate different substances and that type of thing. We'll get to that in a second. So hopefully that makes sense. We'll just go back here. So enzymes, you have the enzyme, and then you have the thing that they act upon, which is called the substrate, okay? So that's important to remember. And basically these enzymes, they can physically orientate the substrates for them to react easier. They can strain them as well, and they can add electric charges as well. So that's something that you'll just have to remember. So how enzymes and substrates actually bind together. So remember, enzymes are 3D proteins, okay? Each enzyme has something called an active site. Now, this active site is a space into which the substrate fits into, okay? And it's at that level or that spot where the enzyme works upon the substrate, okay? Now, this is something you do have to remember, is that substrates are held to the enzyme by van der Waals forces, hydrogen bonding, electrostatic forces, and hydrophobic interactions. You might get a multiple choice question on it, probably won't, okay? The important thing to realize is that enzymes, most of them are reversible, okay? So there shouldn't be any irreversible binding, okay? So if you remember from chemistry, remember that covalent bonds are very, very strong, and they're quite hard to overcome compared to hydrogen bonds, which are pretty easy to break apart, okay? And you should remember this from double-stranded DNA as well, how hydrogen bonds, they're in between the DNA strands and they're really easy to pull apart. Same thing here, okay? You don't have to know in detail what a van der Waals force is, okay? You just have to know that that's there, okay? So we have two different models of enzyme binding. So I have a bit of a diagram here. The first sort of theory is called lock and key. So if we have a look at this diagram here, you can see the substrate and you can see the active site. Now you can see the active site is exactly complementary to the substrate, okay? So it just fits in really nicely there. Now, this is compared to induced fit, where you can see the substrate here, but the active site isn't exactly complementary, okay? So what happens in induced fit is that it sort of wiggles into the enzyme, okay? So the enzyme can change its shape very slightly to help it fit in. Again, this is something you just have to memorize. It's pretty self-explanatory almost, but you probably won't have any questions on that. This is quite important, and you might get an exam question on this. So factors that affect the amount of enzymes, okay? So firstly, enzymes have different lifespans. So some enzymes are only around for a very short period, and others are actually around pretty much for your entire life, okay? So it's just going to depend how old they are. Synthesis of enzymes can be induced or repressed by other molecules, okay? And we're going to come back to this in a second, and we're going to talk about positive and negative feedback loops. We're going to go more into detail about this. But basically, sometimes you can have more or less enzyme depending on other molecules that either block their synthesis or increase their synthesis, okay? Another important thing to realize is that synthesized enzymes are God, I can't speak English anymore, compartmentalized. So basically what this means is that sometimes in certain muscle tissue or in certain parts of the cell, there'll be more enzyme in that specific area. There's an example that I was thinking about putting in. It might confuse some of you, so I'll just say it now. So there's an enzyme called creatine kinase, which you'll come across a bit later in metabolism, okay? And this specific enzyme is used in anaerobic respiration when we need a lot of energy, okay? Maybe when we're sprinting or something like that. And so it makes sense that this enzyme is really localized in skeletal muscle, okay? So there's a lot of it there compared to, say, the brain, which is not going to need it. Okay, so that's why it's important to realize they're compartmentalized. There's just another thing here called isoenzymes, and you might come across this as well. So an isoenzyme is a different enzyme, okay? So if we look at the amino acid sequence, they're different proteins, but it can catalyze the same reaction as another enzyme. So they have the same function, but they're different form. And you just have to remember that. This is also quite important, rates of enzyme reactions. And this you very well might have an exam question on it. So listen up to this. We're just going to skip to the next slide, which has a diagram of this. So basically, enzyme rate of reactions depend on different things. So first, if we have a look at enzyme concentration here, it makes sense that the more enzymes you have, the higher the rate of, or the greater the rate of reaction, okay? Because there's more enzymes and there's more substrate enzyme complexes being formed, so it speeds up the rate of reaction. However, as you can see, it goes up to a point and it sort of flattens out, okay? There's a point here, it's called the point of saturation, all right, where all the enzymes are sort of occupied, okay? And adding more enzymes will not increase the rate of reaction at this point, okay? Because there's a limit to the amount of complexes that can be formed. This is also kind of similar to substrate concentration. So again, if you're increasing the amount of substrate, okay, the rate of reaction is going up, again, up to the point of saturation, where it no longer increases, okay? And that's because all the enzymes have been occupied, okay? So that's pretty important to remember. And you very well might get given a graph like this to sort of understand. Again, none of this is memorizing. You should just be able to look at the axes, okay, and just figure out what's going from there. Now, temperature is a really important one, okay? So, for example, we have something called optimum temperature. So every enzyme will have an optimum temperature where it just works the best, okay? And it'll have its biggest activity at that level. Now, if we increase the temperature, okay, you can see that the activity of the enzyme starts to drop. Oh, we'll go on to the next slide. Starts to drop to a point where it no longer functions. So here, if we were at this temperature, it would no longer work. And that is called enzyme denaturation. You should probably know this from bio and chem. Okay, and that's because all the bonds in the enzymes have been broken at a high temperature. Okay, so that's called denaturation. It's very important to remember. Now, if we look at this side, okay, this is a colder temperature. It still works. It's just going to go slower. So again, you have to think about if we decrease the temperature of any reaction, the molecules are going to start traveling at a less speed, okay? And so they're going to bump into each other less, and that's why there's less rate of reaction there, okay? But even at low temperatures, it still sort of works, just very slowly, okay? Optimum pH is also quite important, okay? So again, has its optimum pH where it works really, really well. If it goes either side of that optimum, it starts slowing down, okay? At extreme pHs, whether it's extremely acidic or alkalotic, it might start to denature because again, that pH can interfere with the bonds, all right? So you might get given a graph, something like this, and you have to interpret it. Just going back to temperature, just a fun little knowledge thing. This is why really high fevers in humans is really bad. So you have a fever over 40 degrees. That's really, really bad because a lot of our enzymes in our body function optimally at 37 degrees, and other proteins as well, not just enzymes. Okay, if we increase that to 40 degrees, our proteins, our enzymes start to denature and they start to not function. Okay, so that's why a high fever is actually quite dangerous. Yeah, so fun point. Next, we'll just talk about allosteric regulation. So I know that's a fancy word, but all it means is that there is another molecule that comes in, okay, and binds to another site on the enzyme, which is not the active site, and brings about a change in the shape of the enzyme, and basically making it not able to bind with its substrate, okay? So if we see this picture here, we have an inhibitor here. It comes and binds at the allosteric site, okay, which is different to the active site. It binds, and if we look at this picture here, you can see that the active site has changed shape, okay? It can no longer bind to that substrate, making it that the enzyme can't work anymore. Now, it can also, so there's inhibitors, but there's also activators, okay? So it might bind to an enzyme and might make it fit better with its substrate there, okay? So allosteric regulation is almost always reversible and non-covalent, okay? If it is irreversible, that generally means it's a poison, okay? So, and that will be coming up a bit later as well, and that's important to understand that. So again, allosteric regulation is reversible. So now we're going to talk about feedback regulation, which is basically a type of allosteric regulation. So in your lectures, they'll talk about negative feedback, okay? And there's also positive feedback, but isn't really focused as much upon in the lecture for some reason. So basically, negative feedback is, for example, we'll just look at this example here. Your body temperature exceeds 37 degrees Celsius, okay? Your body senses this change, okay, and it decides to you know, lower that temperature, and then it sends out different signals to the body to bring back that temperature down. Okay, so if we have a look at the previous slide, pretend this is some sort of enzyme, okay, and its body has realized that there's too much of the substrate or the product being produced, okay? So it might send out an inhibitor and it'll link to this enzyme at this allosteric site and might change the active site, and it can't bind, therefore producing less of the product, okay? So it's a type of regulation. So negative feedback means that there is less being produced, whereas positive feedback means there is more of something being produced, okay? Now, enzyme inhibition is also really, really important to remember, okay? And this is definitions that you just need to know. Put them on a flashcard or something like that, and you'll know, okay? So there's two types of inhibition in general. There's irreversible inhibition and reversible inhibition. Irreversible is very uncommon in biology, just because we don't really want to have irreversible inhibition of something, okay? And generally, if it is irreversible, again, it's a poison, all right? So there's different types of reversible inhibition, okay? So we have something called competitive inhibitors. So they usually have the same or a very similar structure to the substrate, and it binds to the enzyme instead of the substrate, okay? And this can be quite easily overcome by adding more of the substrate. So the substrate is more likely to bind to the enzyme than the other inhibitor, okay? Then we have something called an uncompetitive inhibitor. And basically what this does is that the inhibitor molecule comes and binds in between the enzyme and the substrate, and therefore the enzyme can't actually work on the substrate. Then we have a non-competitive inhibitor, which is binding at an allosteric site. So if you go back to this one, this is a non-competitive inhibitor because it's binding elsewhere, okay? So that's all you really need to know for that. I think Izzy actually covered this slide separately, but phosphorylation of enzymes is more specific to enzymes. How I like to remember it is that phosphorylation is catalyzed by protein kinases. Dephosphorylation is catalyzed by protein phosphatases. How I remember this is that protein phosphatases is a longer word, or longer two words than protein kinase, and dephosphorylation is a longer word than phosphorylation. So that's how I like to remember, and that's all you need to know about that, and you really don't have to know anything else. Moving on. Again, this is just more definitions that you will have to know, okay? So there's different types of helping enzymes, okay? So these are other molecules that come and just help out the enzyme with its function. So we have something called coenzymes. So they are organic, non-protein molecules, which basically help the enzyme function a little bit better. And it is not permanently bound. So the words I bolded here, organic, non-protein, and not permanently bound, are very important to remember, okay? So you just have to memorize that. A really good example of a coenzyme is vitamin B, okay? And vitamin B will come up a bit later in metabolism as well. Next thing we have here is called a cofactor. So they're mainly inorganic ions such as iron, copper, zinc, or non-protein compounds. The difference between cofactors and coenzymes is that cofactors are permanently bound. Okay, so they're always going to be present on the enzyme. Lastly, we have prosthetic groups. So they're non-protein groups that just permanently bound to their enzymes. That's it. You might, I mean, it's very unlikely you'd have any questions in the exam on this, but it's just something you have to memorize the definitions of. Now, moving on to cellular respiration. So this should be a lot of revision from Year 12 Bio or the Bio Bridging course with a couple of extra bits here and there. So I'll highlight the ones that you probably wouldn't know from Year 12. So just a bit of an overview. So cellular respiration is a process of converting glucose to ATP. And there's two types of cellular respiration. There's aerobic respiration, which has to happen in the presence of a mitochondria, and anaerobic respiration, which happens in the cytoplasm. As the name implies, aerobic requires oxygen, anaerobic does not require oxygen. And we'll go into detail a bit more later. So in aerobic respiration, there are three main steps: glycolysis, which also happens in anaerobic, citric acid cycle, and electron transport chain. And we'll go into detail a bit more. This little summary here, you probably should memorize, okay? So aerobic happens in the mitochondria, oxygen is required, goes through the three steps, around about 30 ATP produced, and it's quite a slow process. Now, you'll probably have seen different figures for how many ATP is produced. You do not know, you will never be asked how many ATP is produced in aerobic respiration, okay? You just have to know it's a lot more than anaerobic, where there's only 2 ATP produced, okay? So you should learn the differences, in the body, so we mainly use aerobic respiration, but anaerobic respiration is also used to sometimes, for example, if we're running really fast and we don't have a lot of oxygen and we need ATP produced really fast, then we might use it there. There's also one other really important place where we use anaerobic respiration, which is actually red blood cells. So red blood cells are a little bit weird, okay? They don't actually have like many organelles in them, and they don't have mitochondria. So they just have to rely on glycolysis. Moving on. So glycolysis is the process of converting glucose to pyruvate, okay? So see here we have the inputs of glycolysis and the outputs. This is quite important to learn, okay? I really recommend learning it. Again, you will not have a question being like, how many NADHs are produced in glycolysis? You will not. But it's still important to realize that NADH is produced because there are other metabolic processes that can impact the levels of NAD+ available. If you don't have a lot of NAD+, you will not be able to convert to a lot of NADH. So it's still important to know the outputs and inputs, okay? So if you see here, we have a diagram. So we have glucose, which is a 6-carbon molecule, okay? And it goes through all of these processes to produce pyruvate. It's important to note 1 glucose makes 2 pyruvate molecules, okay? So you can see it's basically split in half, okay? Because pyruvate is a 3-carbon molecule. You do not have to know all the names of these intermediate molecules, okay? If you really want, you can, but really I'd advise against that, to be honest. And if you did want to learn one, you'd focus on fructose 1,6-biphosphate, okay, if you really wanted to learn that. And you can see here, it says it's unstable, so it really does not like existing in this form. It really wants to go and be converted into pyruvate. Okay, so here's really important, particularly focusing on NAD+ and NADH. in biology and chemistry, you would not really have had much focus on NAD+ or NADH, but in medicine for cellular respiration, we really care about these levels, okay? So just important to know in glycolysis, we have 2 NAD+ converting to 2 NADH, okay? So just remember that. Then we have something called the link reaction, okay? So again, in biology, it gets a bit confusing because you might have learned that glycolysis is glucose to acetyl coenzyme A, which is actually wrong. Glycolysis is glucose to pyruvate. And then there's a separate reaction called the link reaction, which converts pyruvate to acetyl coenzyme A. So some people have learned it a bit differently in high school, but this is how we learn it in medicine, okay? Main thing to remember here is that carbon dioxide is released, okay, per pyruvate. And again, NAD+ is converted to NADH2, okay? So that's really important. That's all you have to know here, pretty much. Yeah. Oh, and it happens in the mitochondrial matrix, okay? That's just another, this is for aerobic respiration, okay? Yeah, so this is just a diagram. Again, you just have to remember the inputs and outputs. Really super duper important thing to remember here is the enzyme, which this reaction requires. And hint, there might be a question coming up on this later in this lecture, so please memorize this. So pyruvate dehydrogenase is needed to convert pyruvate to acetyl coenzyme A. And for this enzyme to work, you really need thiamine, which is also known as vitamin B. And if you remember, vitamin B is a coenzyme. So this is a good sort of example of what a coenzyme is. And it helps pyruvate dehydrogenase work. Okay, so that's just an important thing to remember. Now, there's also a link reaction in anaerobic respiration. So this takes place in the cytoplasm, okay? And it converts pyruvate to something called lactate, okay? We also call it fermentation as well. The enzyme required here is lactate dehydrogenase. So it sort of makes sense because here pyruvate to lactate is the one being produced, so it's lactate dehydrogenase. Okay, so that's how you remember it. Whereas this is pyruvate dehydrogenase because it's pyruvate to acetyl coenzyme A. Okay, so this happens, okay, if there is a lot of glucose that isn't being stored or converted further into acetyl coenzyme A. Okay, so this is actually not really good because sometimes we can have a buildup of lactate, okay, which is actually quite acidic and it can cause something called lactic acidosis, okay. So this is where pH becomes important and it causes a low pH. And we treat this with sodium bicarbonate, which you should remember from chemistry is a weak base, okay. So this is important and you very well might be asked a question in the exam about this. How do you treat lactic acidosis? All you have to know about this is that this reaction, pyruvate to lactate, is actually favored by a high concentration of NADH+. This will become super important when you go over alcohol metabolism because if you drink a lot of alcohol, you're actually producing a lot of NADH+. And because you're producing a lot of NADH+, it's powering this conversion from pyruvate to lactate, okay? So in the future, you might actually be treating patients who have drunk too much alcohol and are in lactic acidosis, okay? So that's why it's important to remember this. Okay, now we have something else called the pentose phosphate pathway. You might come across this a bit later. I think it's the last metabolism lecture that you'll have where you'll cover this. So all you have to know is that it's a bit separate to glycolysis. Sometimes something happens in the body where they're not able to convert glucose to coenzyme A through glycolysis. So instead, it just goes through the pentose phosphate pathway, okay? And actually happening quite often in our body, as much as 30% of our glucose breakdown in the liver and fat cells is accomplished by this pathway. That's not something you have to remember, you just have to know it exists. And that's honestly all you have to know is that it's just an alternative to glycolysis if it can't happen. Citric acid cycle, also known as a Krebs cycle, happens in aerobic respiration only. So its whole purpose is to generate a bit of ATP, but mainly to generate NADH and FADH2 from acetyl coenzyme A, okay? So again, you have to learn these inputs and you have to learn these outputs. Yes, that's something you definitely have to remember. And really focusing that it's producing NADH2 and FADH2, okay? That's super duper important. So I have a bit of a diagram here If you really want, you can learn all the intermediary molecules here. It's really unlikely they would ask you, be really super mean of them too, but if you really want to, you can. Main ones to probably focus on is citrate, alpha ketoglutarate, and oxaloacetate. Those are probably the main ones I would focus on if I were you and really wanted to learn this, okay? So one important thing to note is a lot of NAD+ will favor this cycle, okay? Because as you can see, NAD+ is being converted to NADH. If there's more NAD+, there's more NADH being produced, okay? So that's really important to remember. Also here, might be a little bit confused, and this is a bit different to year 12 biology chemistry, is the difference between ATP and GTP, okay? So ATP is our main energy currency in the cell, and that's what we want to produce with cellular respiration. GTP is also something that can be used as energy, but it's actually used more as a signaling molecule in the body. That's why we prefer ATP than GTP, but it can also be used for energy, so don't get too confused about that. Now, moving on to the electron transport chain. This is where it gets a little bit confusing, and we go into a bit more detail in medicine, like a little bit more detail than year 12 biology. But we'll have a go. Okay, so here we have a diagram. This is a really good diagram, except for the fact that they forget to put in an extra membrane. So if we have a look at the next slide, you can see here, this is on the membrane of the mitochondria. So remember, it has a double membrane, okay? This here is the inner membrane of the mitochondria, and this here is the outer membrane, okay? And this here is called the matrix of the mitochondria. So just keep in mind that there's also another membrane here. This space is called the mitochondrial intermembrane space, okay? Because it's between the membranes. So as we look at this, just remember there is a membrane up here. All right, so what happens? Remember, we've produced a lot of NADH and FADH2 from the citric acid cycle. So what happens is NADH, so firstly, what NADH and FADH2 are, they're called electron transport carriers. So if you remember back to your chemistry, remember a hydrogen atom is basically a proton, so it's just one electron, okay? And NADH just carries that hydrogen to the electron transport chain. Okay, so the electron transport chain consists of 4 protein complexes. So you can see 1, 2, 3, and 4. Okay, so these are just proteins embedded in the membrane. What happens is NADH, the carrier molecule, comes and releases its hydrogen through this complex 1. Okay, so it's going from the matrix of the mitochondria into the intermembrane space, and it's leaving its hydrogen proton there, okay? That NADH then becomes NAD+, okay, and it's ready to go back and take more hydrogen and repeat this cycle. FADH2, okay, really likes complex number 2, okay, so that's where it binds to and it releases its hydrogen, okay? Now, as you can see here, complex 2 doesn't directly release hydrogens into the intermembrane space, okay? It actually travels through a different molecule and then goes to complex 3 where it can release its hydrogens, okay? This molecule is actually called ubiquinone. So on the slides later, you'll see there's a definition for that. And all it does is that it transports the hydrogens from FADH2 to complex number 3, okay? Now, complex number 3 actually keeps, so all throughout this, there's hydrogens being released from complex 1 and complex 3, but they also keep traveling along to complex number 4. Now, complex number 4 is really, really important because this is where oxygen acts as the final electron acceptor, okay? That's all you have to know about oxygen, is that it accepts electrons or hydrogens, okay, and it forms water. Okay, so basically the whole purpose of why we have oxygen here is to create a hydrogen gradient, okay? So if you see here, there's so many more hydrogens in the intermembrane space than there are in the matrix, okay? So there's only a couple here. Now with the matrix, sometimes there's hydrogens just floating around in the matrix, okay? And we want to get rid of these hydrogens because we want to make a bigger concentration gradient between here and here. Okay, so we want to get rid of these hydrogens. And how we do that is we combine these hydrogens with oxygen to create water. Okay, and that's an important output you have to know, is that it creates water. So that's why there's a big hydrogen concentration gradient here coming from the matrix and the electron transporters, and because there's less hydrogen in the matrix. Now, why we have a gradient concentration here is to power this other protein here called ATP synthase. Okay, so you should know, or I should have mentioned before, anything ending with the with ase is an enzyme. Okay, so synthase, lipase, those are all enzymes. Okay, so ATP synthase means that it's just building ATP. Okay, and how it works is that all these hydrogens come in through this enzyme, powers this enzyme, and creates ATP from ADP. Okay, so I'll repeat that one more time. So from this big concentration gradient, they flow through here because they don't like being in spaces where there's a lot of them. So it flows through here and it powers the enzyme to make ATP from ADP. Okay, so you might also see that all of this is referred to as electron transport chain, and this specifically is called chemoosmosis. But yeah, that's just for fun if you want to know that. So that's just that. With this, you don't really have to know too much detail about it. Just know NADH likes complex 1, FADH2 likes complex 2, okay? 3 just transport electrons, and number 4 is where oxygen acts as the final electron acceptor, okay? How I like to remember it is FADH2 has a 2, so it likes complex number 2, okay? And you can remember NADH is just the first one. Yeah, so this just goes into detail what I just said. That's pretty much all you have to know about it. This just, if you want to learn more about it, you can, but that's pretty much all you should know. So one last thing, there should be an active learning session that you guys might have about poisons. It was really confusing and it's kind of gross. At the end of the active learning session, you'll be actually given this summary, okay? So all it focuses is that poisons affecting cellular respiration, okay? Technically, anything in an active learning session is examinable. However, this is very unlikely to come up on the exam, and I don't think anyone has ever had this on an exam. But if you want to know it, you can still go through it. I won't be going through it today because it's quite low yield, okay? But if you do want to focus on one, focus on cyanide, okay? Yeah, and just know that that one inhibits cellular respiration, particularly inhibits the oxygen, okay? So it actually binds to oxygen and it doesn't let it accept the hydrogens, therefore not creating a concentration gradient to produce ATP. So that's pretty much all you have to know. So if you have any questions, please chuck them in the chat. Otherwise, we'll move on to some questions that I have here. So these are a bit difficult. Yeah, so oh, maybe not, who knows? So question 1, what would occur with a vitamin B deficiency? Okay, so we had a couple of answers. So it's actually a buildup of lactate. Okay, so this is very well maybe asked on an exam, like what would occur with the vitamin B deficiency. This is quite similar. So it's a buildup of lactate. Okay, so remembering that vitamin B is a key enzyme necessary for pyruvate dehydrogenase, which converts pyruvate to acetyl coenzyme A. Okay, if you don't have vitamin B, okay, pyruvate dehydrogenase cannot convert pyruvate to acetyl coenzyme A. Now, pyruvate wants to be converted, so then it ends up being converted into lactate. So that's why there's a buildup of lactate. This is pretty tricky, I know, and you just have to remember pyruvate dehydrogenase is vitamin B, okay? That's the main thing to take from there. But that's a tricky question, I know, we just covered that just then, so don't worry. Okay, question number 2. Inhibitase is an inhibitor of pyruvate dehydrogenase. If there was an inhibitase present, there would be. So this is an example of what one of the poisons can be presented in the exam. So if we go back to our diagram here, why I recommend not learning that is they'll probably give you inhibitase and they'll describe what it does, okay? So, for example, they'll give you inhibitase and they'll describe what it does. So it inhibits pyruvate dehydrogenase. If you know what pyruvate dehydrogenase does, you should be able to know what happens, okay? So have a go. It's okay if you get it wrong, it's fine. All right, so it's a decrease in ATP, okay? This is a tricky question, all right? So you have to remember what pyruvate dehydrogenase does. So it converts pyruvate to acetyl coenzyme A, okay? If this enzyme is inhibited, there is no acetyl coenzyme A being produced, so there is no citric acid cycle, okay? So as a result, since there's no citric acid cycle, there won't be any electron transport chain, which is where the majority of our ATP is produced, okay? So moving on. Again, this is sort of an example of why it's important to know NAD+ and NADH in all of cellular respiration. So an excess NAD+ leads to what excess buildup? This is a bit of a trick question, by the way. Just have a go. Okay, so yes. Yep, well done. People have got this correct. Really, really good. So we have to remember that NAD+ is used in the cellular reaction for the link reaction, in aerobic respiration. Okay, so that's when pyruvate gets converted to acetyl coenzyme A. So you can see here that NAD+ is being converted to NADH in this link reaction. If you also go back to the citric acid cycle, you also have to remember that NAD+ is converted to NADH. So there's more NAD+, there's more NADH, so it powers the citric acid cycle more, okay? Question number 4, how is lactic acidosis treated? Yep, well done. So I see some A's. Yep, that's correct. So just have to remember sodium bicarbonate is a weak base. Kind of ran out of multiple choice answers at the end. I was doing this at 3 a.m. in the morning, like a week ago. So I was really tired. So yeah, so just remember that sodium bicarbonate treats lactic acidosis. Okay, so I'm pretty sure that's it. Anyone have any other burning questions to add? Feel free. Okay, so it kind of seems no one really has any questions. Feel free to use the Curious Cat or message me directly on Facebook. I really don't mind. Self-isolation is a bit lonely. So please send me questions. Next week, stay tuned for metabolism, okay? We'll be covering all of it. I know this week will be really tricky. And if you have any serious questions, please feel free to message me and I'll try and answer them. And if everyone's inundating me with a lot of messages, I might make a concept video